Hey guys, and welcome back. So now we're going to go over the lipids section from the biological molecules of the biology revision. So the lipids are another group of organic chemicals that play a vital role in organisms. They form an integral part of all cell membranes and are also used as an energy store. Many plants and animals can convert spare food into oils or fats to use when they are needed. For example, the seeds of plants contain lipids to provide energy for the seeding when it starts to grow, which is why seeds are such an important food source for many animals. Fats and oils. Fats and oils are important groups of lipids. Chemically, they are extremely similar, but fats such as butter are solids at room temperature and oils such as olive oil are liquids. Like carbohydrates, all lipid molecules contain the chemical elements carbon, hydrogen, and oxygen. However, lipids contain a considerably lower proportion of oxygen than carbohydrates. Fats and oils are made up of two types of organic chemicals, fatty acids and glycerol. You don't need to know that. I'm just telling you the structure of glycerol. They are combined using Estebans. Glycerol has the chemical formula C3H8O3. They also won't ask you that. Like, what's the chemical formula? They might ask you to draw it. So if they ask you to draw it, then there it is. Right. All fatty acids have a long hydrocarbon chain, a pleated backbone of carbon atoms with hydrogen atoms attached and a carboxyl group at one end. Okay, so this is what they're talking about. This is the glycerol, and this is the fatty acid, and this is the COOH at one end. Living tissues contain more than 70 different kinds of fatty acids. Fatty acids vary in two ways. The length of the carbon chain can differ. So, although often 15 to 17 carbon atoms long in organisms, the fatty acids may be saturated or unsaturated fatty acids. Okay, so this is not really important. In saturated fatty acid, each carbon atom is joined to the one next to it by a single covalent bond. If you're doing A-level chemistry, you would also come across the saturated and unsaturated carbon bonds. A common example is stearic acid. In an unsaturated fatty acid, the carbon chains have one or more double covalent bonds in them. A monounsaturated fatty acid has one double bond, and a polyunsaturated fatty acid has more than one double bond. Linoleic acid is an example of a polyunsaturated fatty acid. It is an essential fatty acid in our diet because we cannot make it from other chemicals. Okay, so a few things to clear up. Right. This is stearic acid, and they are saturated. So what that means is each carbon has is formed the maximum bonds it can. So any carbon you go to is formed the maximum bonds. It's all formed 4 bonds. A unsaturated, you might see this happening. So a double bond. So therefore, this is how it would look like. So, and it would look like that throughout the hydrocarbon chain. And when a fatty acid and a glycerol is bonded together, the bond created is called an ester bond. In carbohydrates, it was a glycosidic bond, and now we're learning about Estebans. Okay, and they've displayed it here again. Forming ester bonds. A fat or oil results when glycerol combines with 1, 2, or 3 fatty acids to form a monoglyceride, a diglyceride, or a triglyceride. This is what we're focusing on, triglycerides. A bond is formed in a condensation reaction between the carboxyl group of a fatty acid and one of the hydroxyl groups of the glycerol, so again producing water. A molecule of water is removed and the resulting bond is known as an ester bond. This type of condensation reaction is called esterification. The nature of the lipid formed depends on which fatty acids are present. So, for example, lipids containing saturated fatty acids are more likely to be solid at room temperature than those containing unsaturated fatty acids. For simplicity, fatty acids are represented by this general formula where R represents the hydrocarbon chain. The fatty acids below are drawn in reversed form. Okay, so this is why it's called a triglyceride. It's 3 fatty acids that are attached. So again, we have the H2O that's being removed, and they can ask you to draw this in the exam, in which you draw the glycerol and you would draw the fatty acid with the hydrocarbon chain represented as R. This is the ester bond that I've showed you. Sometimes they can ask you to draw it and then point out where the ester bond is. Right, the nature of lipids. Lipids contain many carbon-hydrogen bonds and little oxygen. When lipids are oxidized in respiration, the bonds are broken and carbon dioxide and water are the ultimate products. This reaction can be used to drive the production of a lot of ATP. Lipids, especially triglycerides, store about 3 times as much energy as the same mass of carbohydrates. And that's how we know that triglycerides give you more energy. The hydrophobic nature of lipids is a key feature of their role in waterproofing organisms. Oils are important in waterproofing the fur and feathers of mammals and birds, while insects and plants use waxes for waterproofing their outer surfaces. Lipids are good insulators, so we've established that they are hydrophobic, and that's good to prevent waterlogging. And now we're saying they're good insulators. So a fatty sheath insulates your nerves so the electrical impulse travels faster. You will learn about this later on. They also insulate animals against heat loss, so a thicker layer helps them. The thicker layer of blubber in whales is a good example. Lipids have a because against heat loss, because you don't have blood vessels in your fat. So if you've got lots of layers of fats, the blood vessels are going to be lower down, and so you lose less heat through radiation. Because if this was your skin and that was your fatty layer and these are your blood cells, that's the blood moving, the heat from the blood has to travel all through this to escape, and that's hard to do. However, if you've got this layer of fat, and these are your blood vessels, and you've got the blood travelling, the heat of the blood is easier for it to travel out, and you lose heat by radiation. That's why fat is a good insulator. Right. Lipids have a very low density, so the body fat of water mammals helps them to float easily. Good thing. So low density so they can float. All lipids dissolve in organic solvents. So organic solvent, that will be an alcohol, such as acetone, but are insoluble in water, so lipids do not interfere with the many water-based reactions that go on in the cytoplasm cell. That's good. Phospholipids. Inorganic phosphate ions are present in the cytoplasm of every cell. Sometimes one of the hydroxyl groups of glycerol undergoes an esterification reaction with a phosphate group instead of a fatty acid, and a simple phospholipid is formed. Phospholipids are important because the lipid and the phosphate parts of the molecule give it very different properties. The fatty acid chains of a phospholipid are neutral and insoluble in water. In contrast, the phosphate heads carry a small negative charge and is soluble in water. So we've got a part that is hydrophobic, and we've got a part that is hydrophilic. When these phospholipids come in contact with water, the two parts of the molecule behave differently. The polar phosphate part is hydrophilic and dissolves readily in water. The lipid tails are hydrophobic, so they do not dissolve in water. If the molecules are tightly packed in water, they either form a monolayer with the hydrophilic heads in the water and the hydrophobic lipid tails in the air, or clusters called micelles. In a micelle, all the hydrophilic heads point outwards and the hydrophobic tails are inside. Okay, so this is a micelle. So the tails cluster together so they won't have to touch the water, whereas in a monolayer, all the heads are in the water and the tails will be out in the air. They don't want to touch the water, they will not mix. So a phospholipid monolayer may form at a surface between air and water, but this is a fairly rare situation in living cells where there are water-based solutions on either side of the membranes. With water on each side, the phospholipid molecules form a bilayer with the hydrophilic heads pointing into the water, protecting the hydrophobic tails in the middle. This structure, the unit membrane, is the basis of all membranes. And so this is what your membrane looks like. You've got the phosphate heads in the aqueous solution with the water, and you've got in the middle the fatty acids, and again you've got the hydrophilic heads in the solution. And so this is called a bilayer. Bi meaning two, and that's two layers formed. And that's it.